What is optogenetics and how does it work? Optogenetics is a method of controlling and monitoring the functions of cells, tissues, organisms by combining specialized optical systems and genetic engineering technologies. It is now considered a branched biotechnology because of its wide use in studying the brain's neural circuits. Optogenetics is an evolutionary approach without which understanding brains wouldn't have been possible. The technique allows researchers to turn a specific cell or cellular region on and off with precision and high resolution by using a protein called opsin, thus enabling them to control animal-specific behavior, such as pain and fear, and demystifying the involvement of specific cells in controlling those behaviors. Francis Crick first proposed the concept of optogenetics in 1979 that light can be used to obtain rapid space-time dependent control over targeted neurons. However, the ideas wasn't applied in neuroscience due to the unavailability of advanced tools and methods. Optogenetics has broad applications in neurology, animal behavior, and physiology to provide insights into a spectrum of questions related to movement, sleep, blood pressure, memory, hunger, body metabolism, fear, learning, navigation, and sensory processing. I want to point out this word opsin is also include the word sin in there, SIN. They say they don't have the advanced tools and methods. It's an excuse to stop people from speculating what they've been experimenting. So technically, they have everything to experiment on human brains, okay? And of course, animals. Key tools involved in optogenetics. There are three tools to perform targeted expressions and studies. Number one, opsins, or light-sensitive proteins. Researchers require a light-activated ion channel or pump, such as opsins, to provide targeted control over electrical and biochemical events of a cell or specific regions at a particular time. Types of opsins. Opsins have the unique ability to sense and respond to light. It's categorized into two groups based on its mode of action and primary sequence. Type 1 are microbial opsins. They are found in microorganisms such as fungi, algae, archaebacteria, and eubacteria. They include light-driven ion pumps like ion channels, channel rhodopsins, bacteriorhodopsin, halorhodopsin, and sensors, rhodopsins 1 and 2. Microbial opsins are widely used in epigenetic research because they have faster kinetics. They are single component proteins that can easily be genetically engineered. Type 2 are animal opsins. They are found in animals and humans. Examples of rhodopsin and melanopsin. They couple with G protein-mediated transduction pathways to mediate transmembrane RNA potential. They are also primarily involved in circadian clocks and dim light vision. Okay, so G protein they really like the alphabet G. So like I don't know. It just really anyway. Functions of opsins. Microbial opsins like channelrhodopsins are used to excite neurons. Okay. Engineered potassium channels prevent the generation of action potential during blue light illumination. Here I want to highlight blue light illumination. Ever since the pandemic, there is a rumor about blue light. So I think this is related to the optogenetics because they don't necessarily only need to identify who has the potion, but they want to genetically modify humans. So here you go, blue light illumination. Light-driven ion pumps such as hyaluronidase, fungal opsins, archaerodopsin, and enhanced hyaluronidase are used to inhibit neuronal activities. In mammals, opsins are coupled to specific G protein coupled receptors to manipulate the concentration of secondary messengers, such as CAMP or IP3 in target cells. Suitable plasmid and or viral vector. Researchers developed direct and indirect delivery methods to deliver vectors containing genetically engineered or desired genes. The direct delivery methods involve an electric field, laser pulse, hydrodynamic pressure, and ultrasound waves for gene delivery. The indirect delivery methods include photosynthesizing target regions by transplanting photosensitive cells to targeted regions. Um, so, um, the direct delivery methods we are really um, in in this situation right now because we know that they can send any wavelengths and manipulating the electric field, right? Um, so it's not surprising they've been doing that to us, and specifically to the people that have potions. The nanoparticles and dendrimers are also used in some experiments to deliver the protein in targeted regions. However, it has low chance gene expression and delivery efficiency. The most extensively used gene delivery methods use viral vectors such as adenoviruses, lentiviruses, and adeno-associated viruses. They offer better gene delivery efficiency than plasmid vectors. So we all wonder why all of us constantly getting sick. It's possible that they spreading these viruses to get us sick in order to deliver their desired genes into our bodies, so-called desired genes, right? Okay, number 3, light source. A high throughput integrated fiber optic and light source are required to reach the target cells even deep within the brain. The superficial brain areas can be stimulated by directly mounting optical fibers or LEDs to the animal skulls. However, OLEDs are used to implant in the brain because of their thinness. Um, that's say a lot in the healthcare business industry, I should say. Um, you know, it's just amazing how they keep spreading viruses and get us sick so that we always go to the doctors, and the doctors of course prescribe, you know, medicine to us, but who knows what is in the medicine, and who knows if the virus is like really actually is to, you know, deliver uh, the genetic engineered cells into our bodies and cause us to change our behaviors, our, you know, functionings in our bodies, like emotions and etc. How do neurons communicate with each other? Neurons communicate using a combination of electrical and chemical activity. We can use electrical signals to turn some neurons on and see what happens next. This is exactly what scientists started to do. It wasn't until the 1930s that electrical stimulation was used to map the human brain. Dr. Penfield lowered a small electrode into the motor movement areas of the brain. Then he sent a small electrical signal and observed the patient's movements. Stimulation in one area of the brain caused the finger to twitch, while stimulation in a slightly different area of the brain caused a foot twitch. This led Dr. Penfield to realize that certain areas of the brain control very specific areas of the body. Since 1930s, brain stimulation experiments have changed. Electrical stimulation studies have some drawbacks. One problem is that the brain might be damaged when an electrode is inserted. Another problem is that electrical stimulation activates tissue in a very general, non-selective way. It is like using a bulldozer when a shovel will do. The bulldozer is effective, but it is not very precise or careful. In 2005, a new technique was created to allow for more precise brain stimulation. This technique is called optogenetics. Number A. Electrical stimulation. Number B. Light stimulation. Number C. Optogenetic stimulation. So, it's more specific by using optogenetic stimulation. In figure 1A, in an electrical stimulation study, all cells close to the stimulation site will be activated. B. Blue light does not normally affect neurons. Number C. Blue light will selectively activate neurons with channel rhodopsin.